Nasim works with patients and their families as a genetic counselor who come in for genetic testing and for counseling. Genetic testing raises complex ethical issues. What genes do you test for? What do you find? What do you do if you find something you weren't looking for? Who do you tell about this genetic information? Which members of a family have the right to know? Nasim and her genetic counselor colleagues deal with these questions every day.
Hello, everyone. Thank you. Thanks, Kathy. So I work for the Center for Genetic Medicine at Sick Kids, and I'm just going to talk to you a little bit about the clinical impact of genomic medicine or personalized medicine. And I'll show you a case, an example, and hopefully that'll make things sink in a bit more. Okay, so just to review, you probably know this, but we have billions of cells in our bodies. In each cell, we have a library of genetic information. And you can think of this genetic information as a kind of a storybook. So we have about 20,000 stories, and they're packaged into these books called chromosomes. So the DNA, or the genetic information, is coded in 4 letters. C, A, G, and T. And if you look at any location in the genome, people will have different letters at that location. So if you look at that picture with the 3 people, so at the 4th letter, someone might have an A, another person might have a G, another person might have a T, and that's just normal. That's part of being a human. But when we talk about genetic and genomic testing, essentially what we're doing is we are taking a person who has a genetic condition, and we are looking at their genes, and we're trying to figure out, is there a mistake in the genes that would explain what they have? So if I have a child with a heartbeat problem, what we're trying to do is we're trying to look back at one of their 20,000 genes and figure out, is there a mistake in one of those genes that explains the heart problem? So up until about 2000, the year 2000, when we talked about genetic testing, we were looking at 1 gene, 2 genes, 3 genes, maybe a handful of genes for each person. And that allowed us to provide an explanation or a diagnosis in about 10 to 15% of cases. So when you say 10 to 15% diagnosis rate to a genetics person, that's great. To any other person, that sounds pretty horrible. Since the year 2000, there has been a revolution in the technology that allows us to look at most or all the genes at one time. So that cuts down on the cost of genetic testing and the time it takes to do a genetic test. So since then, our diagnosis rate has gone up to about 30 to 50%, which is a huge jump considering where we were to begin with, but we're still not anywhere near perfect. So when we talk about looking at all the genes or most of the genes at one time, that's what's called an exome or a genome test. So you probably have heard those terms. So when we say a genome, that's what we mean. We mean looking at all the genes in one go. That doesn't mean that we can interpret all the genes, though, or interpret what we find in all the genes. Okay. So there are a lot of misconceptions and unrealistic expectations associated with this kind of testing. So you would think if we look at all the genes, why can't we diagnose everyone who has a genetic condition? And some of that has to do with the limitations of the technology. Most of it has to do with our limitations in terms of analyzing and making sense of what we find. So we have about 20,000 genes, each of us. We have 2 copies, 1 copy from mom, 1 copy from dad. Out of these 20,000 genes, we only know what about 3,000 to 5,000 of them do. So the rest of the genome is a sort of a mystery. So we're not sure what those genes do. We're not sure what changes in those genes would do. And a big part of sharing data and doing research is really trying to hone in on, can we make sense of these genes if more than one researcher in the world is looking at them and sharing the data that they find in those genes? Okay. Right. So this pie chart represents a genome. So let's say each colored dot represents a change in the genes. So if you look at any person, if you look at you or me, and we do a genome sequence, we will find millions of changes in our genes. We call these variants. And 99.9% of these changes are what we call benign. So they're normal. They're just part of being a human. That's why I might have curly hair, you might have straight hair, one of us will have blue eyes, another one will have brown eyes. So they don't really affect our health. But there is a portion of these genes where changes will cause disease. So let's think about a child who has a heart problem, for example. If I see this child and we do a genome sequence, we'll find millions of changes in his genome. Most of these changes, if you look at that gray upper portion, most of these variants are in genes where we have no idea what these genes do. So we can't really say anything about them. There will be a small proportion of those changes that will be in heart-related genes, though. So that's promising. That's what we're looking for. Why does he have a heart problem? So those are variants or changes that we would like to follow. Then there's another portion of these variants that are in genes that are important for drug reactions. So is he going to respond well to warfarin, for example? Or if he ever develops cancer, should he be given drug A or drug B? And then there's another section of those changes that are in genes that have nothing to do with the heart, but predict risk of other diseases. So for example, you know some genes are important for heart function, some genes are important for brain function, some genes are important in cancer development. And when you do a genome, essentially you're looking at all of those changes. So you might find something that is related to the heart. You might also find something that has nothing to do with the heart, but is important for that person's health. So this is sort of where the frontier of clinical genetics at this point, where we have all this information and we can tell people about the risk of future disease, but we're struggling with, should we tell? Who should we tell? And what does that mean for the healthcare system and that individual if we tell them, well, in 10 years, you have a high risk of X, Y, or Z? Okay, so here's the case. So we have a 7-year-old boy with a history of that heartbeat problem, and he also had seizures. So we did genome sequencing, and we figured out why he has the heart problem, but we also found that he has a change in a gene called BRCA1. This gene is important in risk of breast and prostate cancer. And usually people don't develop breast and prostate cancer until they're in their 40s, 50s, 60s. So essentially what we're doing is we're predicting risk of future disease for this child. And if you think back to any genetics that you've had, we also inherit our genes from our parents. So if this child has a risk of breast or prostate cancer, what does that mean about his parents? That means that one of them potentially has the same risk. So then the question is, do we tell the child at 7 years of age that when you reach 40s or 50s, you have an increased risk for prostate cancer? Do we test the parents and tell them, well, one of you has a risk of breast cancer, for example? That will be important for the health of that parent, because let's say his mother has the BRCA mutation, then she would need to get mammography, MRIs, and we could potentially prevent breast cancer in her. But that would mean that the whole family now knows that there is a mutation in this child that would give him an increased risk of cancer 30 years down the road, and his siblings might be at risk, his parents might be at risk. So what does that do to the child? Is he going to be worried for the rest of his life? Is that going to affect the way he plans his life? Is that going to affect the way his parents view him? So these are the issues, some of the ethical issues that people are dealing with this kind of testing. It's great, it's really wonderful that we can give this much information out, but not everybody wants that information. And when I talk to patients, some people want to know everything. Some people don't want to know anything other than, why do I have a heart problem? And some people have more nuanced opinions where they say, well, if it's cancer, I want to know, but if it's risk of Alzheimer's, maybe I don't want to know that. And there's also the issue of insurance discrimination. So if you tell a 7-year-old he has a risk for prostate cancer, and then at 20 he wants to get life insurance, what is that going to look like? He's going to have to tell he has this risk. Does that mean he can get insurance, or does that mean he can't get insurance anymore? So these are some of the issues that are that we're dealing with right now, and there's really no right answer. So as you develop and you go into your careers, these are some of the things you might want to think about if you're interested in genetics. And I'm done. Thank you.